All right. Well, good morning. Thank you so much for being here so early in the morning. I really appreciate your presence. And I want to thank the organizers for inviting me here today to tell you about some of the work that's going on in our lab on gene editing for DMD, which stands for Duchenne muscular dystrophy. So the first slide here is my disclosure statement. I'm a consultant for Exonic Therapeutics. So skeletal muscle is one of the largest tissues in our body. It comprises about 40% of our body mass, and as we know, it facilitates our movement, and it's a major site of metabolism, and it's highly regenerative, and we use it every day, so it's very exciting tissue to be studying. And so one can imagine, and it's not surprising, that when there are mutations, genetic mutations that affect muscle, it could be very devastating to our body. And so today I'm going to tell you about such a disease, which is Duchenne muscular dystrophy. So Duchenne muscular dystrophy, it occurs about 1 in 5,000 boys, and currently there's not any effective cure for it. These boys are born seemingly normal, and it's usually the parents that first notice that they're having some trouble walking, and they take them into the pediatrician. And if they are diagnosed with Duchenne muscular dystrophy, most of the boys have the following course. So usually in their early teens, they are wheelchair bound, and then they start to have limited use of their arms. They have ventilation problems, breathing problems, are on ventilators, and usually they succumb to death in their early 20s, early to mid-20s, due to cardiomyopathy. So it's a real devastating disease, as you can see. Now, we have known for 30 years what causes this disease, and it's a mutation in a gene called dystrophin. Now, dystrophin is one of the largest genes in our body, 2.2 million base pairs, and it is comprised of 79 exons, and it is an X chromosome. So the dystrophin gene encodes a protein called dystrophin. And if you look at skeletal muscle and you make a cross-section of a muscle fiber, and you stain it with an antibody to dystrophin, you see this beautiful pattern that is shown here on this slide. And what it's showing us is that dystrophin is actually expressed in the membrane of the muscle. And if you look at this next illustration, it shows you a couple of things about dystrophin. One, it binds to many different proteins in the muscle. But what it actually does is it tethers the cytoskeleton part of the muscle with the extracellular matrix. And we think of it in a way as a muscle shock absorber. So it is intricate and integral for the muscle, and it's important in keeping the integrity of the muscle protein. Now, boys with Duchenne muscular dystrophy, DMD, do not have dystrophin in their muscles. Now, it has been documented that there are greater than 3,000 mutations in the DMD patients. And these mutations come in different forms. There are exon deletions, point mutations, and duplications. So we wanted to attack this disease using CRISPR-Cas9. And so we kind of sat around a table and we said, do we have to come in there and get 3,000 different guides to be able to go in there and make mutations? But that is really not the approach that we wanted to take. So the first thing we decided to do was to take a look and see, well, are there places within this gene that have more mutations? So what I'm showing you here is if you take the exons, the 79 exons, and you display them here on a plot, and you graph them against the exon mutations, what becomes very evident is that there are what we're calling hotspots, meaning that there's parts in the gene that are more susceptible, if you will, to mutations. And so we decided to study these areas that have the mutations. So the dystrophin gene actually lends itself very beautifully to gene editing. And the reason that I say this is it is somewhat of a modular protein, meaning that you can excise parts of this, and it could be a truncated protein, but if it comes back into frame, it in fact will be functional. And that's kind of the approach that I'm going to tell you about now. So if you take again those exons, those 79 exons, and lay them out here, you can see actually we have them color-coded to represent the areas that they code in this protein. And if you look and overlay the exons that we're calling the hotspots here, you see them here in red. So these are the areas of the genes that we've put a lot of effort into, and we've designed guides here to be able to do gene editing of this gene. And earlier in this meeting, Leonela Amoasi has given some talks about exon 50. And today, I'm going to tell you some of our newer work on exon 44 deletions. So one of our goals when we wanted to do gene editing in the dystrophin gene is we wanted to edit as little as possible, kind of keep the genome intact, if you will. And our other approach was we didn't want to come in there with two guides. We wanted to use just one guide. So our goal was to use a single-cut genome editing to restore dystrophin expression. And what became apparent to us and what kind of guided us to where to make the guide is the PAM sequence of the SpCase9. So if you look at that sequence, it's NAG or NGG. And in fact, looking at the splice acceptor site or splice donor site, we see in fact the PAM sequence is there. And so kind of our approach in doing and designing these guides was to design them using the PAM sequence that is the splice acceptor or donor site, depending on where the mutation is in which exon. And then we would be able to kind of eliminate the exon by just destroying the acceptor or donor sites. So that's the approach that we took. And so we tried to use a single guide with all of our editing. All right, so here is kind of an example. What you see here is exon 42 to 46. And I draw to your attention that we've have them illustrated in here in certain shapes, so you can tell whether or not when they come together, spliced together, if they will in fact be in frame. So we are studying one of our patients that actually has a deletion of 44. And without 44, then 43 will splice into 45, and then there is a stop, and so you will not be getting dystrophin being made. However, if we remove 45 and it no longer is an exon, then 43 can splice into 46. You can tell by the shape that it will be, the open reading frame will be reinstated, and we will get dystrophin being made. It will be a truncated form, but it will in fact be in frame. So this we call exon skipping. But I want to introduce one other concept, and it's what we call exon reframing. And by this is the following. So if it is not removing the exon, but actually inserting one or deleting two nucleotides, what in fact that also does is it puts it back in frame. And so how can this happen? Well, we're using NHEJ, non-homologous end joining, because we're using only one guide to do the cutting. And so you get indels actually as the products. So this was also a possibility. And in fact, what happens is that in fact the exon acceptor site is retained, but you get the one insertion or two deletions. And in fact, you will see that you have exon reframing. All right, so how do we interface with our patients? Well, they come into our clinic, and we draw a sample of blood, about 8 mils of blood, and from that we're able to make induced pluripotent stem cells. And we use these cells to do a lot of our gene editing in there. And so we use different types of guides. We try different ones to make sure that we get the best editing for that particular mutation. Once we've accomplished the gene editing, because they're stem cells, we can differentiate them into muscle, cardiomyocytes, and then we ask the question, are we making the dystrophin? I'm going to show you that on this next slide. So here's one of our patients that is lacking exon 44. And so we have one guide that we've designed that will hit 45. And what I'm showing you here is we've done it in the induced pluripotent stem cells. And the panel on the left are cells that have not been edited. And the color it's green, telling me that we have muscle proteins being expressed, troponin. But when we stain with dystrophin, which would show us red, on the cells that haven't been edited, here on the left, you don't see any dystrophin. And on the cells that have been edited with our one guide and Cas9, you can see beautiful dystrophin. This is also shown when we do our Western, and we looked at dystrophin being expressed. And we see here the first lane is our normal dystrophin patients, iPS cells. Here are the non-edited cells, and here's after editing. And so we know that in this dish, we are able to edit and express dystrophin. Now, we have spent a lot of time with different patients and different iPS cells and working this out because it is really critical that we have the right guide to go forward with. But we know in order to get into patients, we have to start and go into animals. And so there is no delta 44 animal for a model. And we decided to go ahead and make one so that we can study it in this organism. So we used actually CRISPR to make this mouse. So in this case, we used 2 guides to cut on either side of exon 44. We removed 44. And as you can see here in immunohistochemistry, on top is a wild-type mouse. You can see TA muscle, diaphragm, and heart, dystrophin being expressed. However, in our delta 44, there is no dystrophin. And again, in a Western blot, you can see in our delta 44 mouse, there is no dystrophin being expressed. Okay, so now we have our mouse model that we want to study. And so we decided to first inject some guide. Actually, we package it up in AAV9. We have our one guide RNA and Cas9. Package it up and do an IM, intramuscular, to see whether or not we can just get it in the muscle when we localize it. I'm going to show you those results here on this slide. So here what you see in the first panel is just a wild-type animal, and you see dystrophin staining, and here are delta 44. As I showed you before, there is no dystrophin. But here we did an injection, an IM injection, and this is 3 weeks afterwards. If you look at the muscle, you can see that dystrophin is being expressed. So our question that we asked is, well, what are we getting at the genomic level? Are we getting editing, or are we getting reframing? And actually, at the time, we were really surprised by the results. So here what we did is look to see what is happening at the genomic level. So I'm showing you here the sequence for the wild-type animal, and then the delta 44, which we don't see. And this is RT-PCR, so we're looking actually at the RNA that is being transcribed. And then the 2 bottom panels here, what I'm showing you is that we're seeing reframing and we're seeing exon skipping. But what was really striking to us is what we saw with the reframing is just an insertion of one. I mean, we always thought that the indels were somewhat, we would call them imprecise, but here what we see consistently and in all the muscles that we've looked at is that we get a reframing with an addition of just an A. And in fact, The reframing was what we saw, was the highest percentage of what we saw. It was about 42% of the products seemed to be reframed. And we only saw 7% actually of exon skipping. So we wanted to go further and take a look to see what happens when we do an IP injection and look at it systemically. So again, we packaged our one guide in with our AAV9 virus, and we did an IP injection very early at P4. And one of the questions we asked, well, how much RNA, how much guide do we need to be adding in there? And so the graduate student who was working on it decided to do increasing amounts of guide to see, do we see any difference in the product if we have more guide? So we kept the Cas9 the same, and what she did here was add increasing amounts of the guide RNA, up to 10 times the amount of Cas. And what was very evident to us, and this is, I'm just showing you here, the TA muscle, is that you get, as you increase the amount of guide, you are getting more dystrophin. And in fact, we saw this in all of the different muscles that we looked at. Here's TA. We also saw it in diaphragm, as well as in the heart and all the muscles that we saw. So this was a really important message to us. It was telling us that it was the guide RNA that was really rate limiting in terms of the editing and in terms of making dystrophin. The other thing that we saw that I just want to point out, and we're still not really sure why this actually is occurring, is when we look to see how much Cas9, even though we put in the same amount of Cas in each one of our injections, what we saw was that with more guide RNA, we in fact saw that we were expressing more Cas9. And at first we thought potentially maybe this had to do with the stability of the Cas9, but in fact what we saw when we looked at RNA was that we saw that there was more Cas9 RNA being expressed. So here is, I hope you can see this, the immunocytochemistry of the various muscles. So this is after one injection, IP injection, and we looked 4 weeks afterwards. What I'm showing you here is in the first panel is the wild type, and we're looking at the TA muscle, triceps, diaphragm, and heart. You can see beautiful dystrophin expression. In our delta, the next lane, the delta 44, you don't see any expression of dystrophin. And as you increase the amount of guide RNA, you can see at 10 times the amount, you see almost all of the myofibers, and the heart is expressing dystrophin. So we also took a look at the whole muscle, and here's what I'm showing you. Is here on the left is our delta 44, where you don't see, this is the TA muscles, and you don't see any dystrophin being expressed. And here is a corrected one, and this was 5 times the amount of guide being used. You can see that most of the muscle is expressing dystrophin. However, at 10 times the amount of guide RNA, we effectively saw that all of the myofibers were expressing dystrophin. So what does the histology look like? Well, we did some H&Es, and in fact, you can tell here that on the left is the wild type histology, and you can see beautiful muscle. And then on the delta 44, you start to see some degradation of the muscle as expected, since there is no dystrophin in these mice. And then after the correction, and here I'm showing you one with 10 times the amount of guide, you see that the muscle effectively looks very similar to a normal or wild type animal. So how about function? Well, we have various assays that we can do, and one of them that I'm going to show you here is actually, it's an ex vivo. So we can take the muscle out and we can measure the force that it is exerting. And here, what I'm showing you in blue is a wild type muscle and measuring the force over time. And here is for our delta 44 animal in red. And then if we look at the muscles of our corrected animal, Here is one of when we've delivered 5 times the amount of the guide. You can see that it is definitely has more force than the delta 44. And here it is again when we're using 10 times the amount of guide. So again, by increasing the amount of guide, we're getting more dystrophin, we're getting better function and rescue of the muscle. So the question is the durability, or how long does the dystrophin in there? I mean, it is great we're doing 1 injection and then waiting over, and we look usually about 4 weeks or 8 weeks, and we're able to see dystrophin. Well, these experiments are still going on. The oldest mouse we have, I'm showing you here, is 6 months old. And we can see we were pretty pleased to find out at 6 months that in fact we're getting quite a bit of dystrophin being made. And here you can see the Western blot showing you of TA, triceps, diaphragm, and heart. And you can see on this Western blot that in fact dystrophin is being expressed 6 months after that 1 injection. So we've gone forward and are made many different mouse models that are representative of the patients that we're seeing. So we've published our Delta 50 mouse, and I've told you a little bit today about our Delta 44. We also have a Delta 52 and a Delta 43 and Delta 45. And we're spending time trying to get the best guides for these models to be able to do our editing. And what we feel, and what I'm showing you here, is that by getting guides for these particular deletions, we'll be able to cover about 50% of the patient population with these guides. And hopefully the next time I give the talk, we'll have more results on how we've done the editing with these other models. So here is the immunohistochemistry, just showing you that in fact we have designed and made these models. You can see here these are the TA muscle, diaphragm, and heart, and these are Delta 44, Delta 52, etc. here. And you can see when you're looking at the immunohistochemistry, they are not making any dystrophin. So we have these models, and this is what we're studying right now in the lab. So here's kind of our vision and our paradigm for what we're doing. We use our patients' blood collections to make iPS cells. We spend a lot of time designing the right guides to be able to do the editing and then check it by differentiating them into cardiomyocytes, as well as we've designed and generated different mouse models of DMD, testing our guides again to make sure that they are effective in the organism. We've done a dog model with the Delta 50, and I believe Leonela Amosi has shown this at the meeting. And that has been working incredibly well. And so, and of course, what our ultimate goal is to be able to deliver these to patients. So some of the real kind of distinguishing features, I mean, what is so wonderful about the gene editing is, in fact, we're going in there, and what we're doing is editing the disease-causing mutation. And we also try to attack this, and our approach is to do minimal changes to the genome and try to use only one guide to be able to go in there and do the editing. In principle, we believe this will be a one-time treatment, and using the AAV9, we're able to reach the skeletal muscle and heart. And because we're editing the gene, what in fact we're doing is still using the regulation of, and we'll get normal expression of the dystrophin, because the regulatory system that is using it is that from the gene itself. So some of the challenges that we still are looking at and we're very aware of is the long-term durability. I mean, we've seen after 6 months we're still getting dystrophin being expressed, but We're looking obviously longer. We have also some injury models that we're looking at to see what happens after the muscle is injured in terms of dystrophin expression after the correction. Also, off targets is a big thing, and we spend a lot of time looking at off targets, as well as the immune response from the virus that we are delivering. So these are the things that we are spending time in and some of the challenges. With that, I'm going to end, and I want to thank and acknowledge many of the people who have been involved, primarily Eric Olson, who has supervised and overseen many of the studies that I am showing here today. Also, Yi-Li Min, who is the graduate student who now currently is working at Exonics, and it is mostly her work that I showed here today. And with that, I thank you for your attention and would gladly take any kind of questions. Thank you.
Turn the switch on, I think.
Okay, yell.
So first of all, that was one of the clearest talks I've ever heard. Thank you. I hope you teach biology. Your students are fortunate. Congratulations on remarkable progress.
Thank you.
I have a question about how you're thinking about interfacing everything you've done with the progress that Leonella described. In particular, your penultimate slide has a mouse efficacy, and you've already built the mice. Then transitioning to a dog, and I just want to make sure I understand how you're thinking about this. The dog efficacy is already done for the one dog that exists.
Right.
I assume you're not thinking about making dogs.
No.
In other words, are you thinking about doing the efficacy in the mice and then doing safety for the human leads in the dog? What experiment do you want to do in the dog? Because you're kind of ready to go to the clinic, basically.
Right. I mean, I think it lies with the FDA what they ask us to do. I mean, we're positioning ourselves in such a way that we're really trying to look at our guide the best that we can, each one of the guides. So really, Delta 50 is what we're going to FDA with initially. And that one we have done in mice. And we didn't make that dog. That was a spontaneous mutation. So we're not making dogs with DMD. And so that, our hope is that if everything goes beautifully with the Delta 50 and we have all of the other guides and we've done all of the work, that the FDA will give us permission to go forward with the other guides. So that's the ultimate goal, hopefully. Hi,
Rhonda,
Jeff.
I wonder if you could clarify some of the points about the durability. Duchenne muscular dystrophy is a lifelong disease.
Yeah.
Are you saying that you feel that if you treat a patient at 4 days of age, they're still going to be making edited dystrophin 70 years later?
Yeah. Most likely not. I mean, but we do want to look at durability over time with our mouse models. So we know that there is turnover of dystrophin, of course. And we know most likely we're not hitting the satellite cells with the virus that we're using right now. So most likely at some point we would have to come back, potentially in some way, and deliver more guide to them. So that we are very aware of that we don't believe, I mean, it would be wonderful that, once we restore the dystrophin that it's there, but we understand that there is some turnover. We are doing some studies. We have some downhill treadmill studies with our corrected mice, and we're looking over time to see, I mean, that's a big question, what happens to these boys when they're out there playing in the playground and there is some injury to the muscle, what happens to the corrected dystrophin? And so we're doing those kinds of studies. So yes, thank you.
Hi, thank you for the wonderful talk. My question is, you made cardiomyocytes from the patient's iPSC.
Yes.
So it would be very beneficial for all the future experiments to have some kind of functional model in the cell. Do you know if it's possible?
Yes.
Yes. Okay.
We do. Actually, it's through collaborations with a group in Germany, Wolfgram Zimmermann, who has these engineered hearts. I didn't show it. I don't know if Leonella MOC showed those pictures, or maybe she didn't, with our iPS cells. We have published it in, I think, Science Advances. We published that paper. So it's a model where, and we sent our cardiomyocytes to them. We also do like calcium transients. We do all of that, but it's actually, they have a functional model of looking at these cardiomyocytes. If you're interested in it, he's a great collaborator, I will tell you. We send him cells and he gets the work done. So if you're interested in that, contact me and I'll get you in touch with him.
All right, thank you very much.
Sure.
Yeah, thanks for the fantastically clear talk. I had a question about the reframing events. So you
Yeah.
showed that you get that one nucleotide insertion. Is that the only indel you see, and what does the diversity of those look like and
Yeah.
frequency?
So it depends where you do it. So when we did it in the cells, in fact, we saw big insertions, deletions, as was expected. So it's interesting. We see this in mice, and we saw it with the delta 50 also in dogs. And it's not all of them. It was, there are some other indels that are there that are not in frame. So if you add up the percentage, it's not 100% of the editing. So there are some that actually edit it not in frame. But for the greater percentage of it, for some reason, there is that insertion of an A. So
Are there other
and-
in-frame events? Sorry, are there other in-frame events that aren't the one nucleotide insertion?
if it is, they're rare. At least with this one. Now, we see it with the delta 50, the same thing, delta 44. With the other ones, it's not all, you don't see it with every single guide. So we don't always see that. I don't know if it's the locus. I have no idea. We're not really sure why we're seeing that, but it is consistent and in all the muscles.
Thank you.
Yep.
I have a quick question.
Yeah.
We have to move on, but so do you care to speculate about why you're seeing more mRNA for Cas?
Yeah. We've thought about this. It potentially maybe that the muscle itself is better, healthier in that way, and so potentially there is more muscle, more Cas, more virus there. I mean, that could be it. I don't know, maybe the muscle that's degrading because it hasn't been corrected. Maybe the virus isn't expressing as well.
So
I
have you
don't
done
know.
any more fine timing studies to see?
Not really.
Okay, yeah.
Not really. It was just, it wasn't something, we were really surprised. When
Right.
At first we thought, well, maybe something's wrong with the Western,
Right.
It took it, and then we thought perhaps it was a stability of the protein, but we see it in the RNA. So really, we're not sure why we're seeing that,
That's
but
great,
it seems
though.
to be consistent.
Okay, thank
All
you very
right.
much.
Sure.